special interest. It's a real pleasure for me to introduce our invited speaker for today, Professor Luca Fontanesi. He is a full professor at the University of Bologna, and its research span a wide range of topics within the animal genetics and genomics, with application also in the authentication and traceability of food products. His work integrates cutting-edge approaches spanning all omics sciences and to better understand and improve the sustainability and animal production systems. His research and scientific interests cover a broad spectrum of species, including the major livestock species, such as pigs, cattle, and rabbits, as well as aquaculture species, wild species, insects, and bees. But also environmental DNA and even extinct animals, a truly impressive and multidisciplinary research. So Professor Fontanesi has been involved in numerous national and international research projects, and he is currently the coordinator of a cross-section, EU-LI-PHE, which aims to build a multidisciplinary and inclusive European network to advance the field of livestock phenomics. Today, he will guide us through a talk titled Phenomics as the central dogma in animal science, linking precision livestock farming with precision animal breeding and selection. So please join me in welcoming Professor Luca Fontanesi.
Okay, thank you very much for a very impressive presentation. And yeah, I'm quite, this is very really an honor for me to be here, and it's a pleasure to be here to present a few concepts derived from the cost action that I'm chairing. Phenomics as a central dogma in animal science is the hashtag that we are launching, developed through the discussion that we had during this couple of years since we started the cost action. I will go through the cost action in a few minutes to represent what it is. Just a few words about where I come from. It's always interesting when you are online to see where people are. I come from Bologna. It is in the north of Italy. Is a middle-aged, well-preserved town with the oldest university in the Western countries. It was founded in the year 1088. And I come from the Department of Agriculture and Food Science that has 10 divisions, and we are working in the animal science division. Of course, there are other divisions that are trying to contaminate our work also. And leading the animal food genomics group, great people that are helping me in what we are doing. Many activities that we developed have been carried out by them with their ideas, with their brains, and novelties in terms of youth, because I'm young at heart, but not yet at age, unfortunately. But anyway, we'll do our best. Okay, so back to the presentation, to the concept.
Sorry,
yeah,
we cannot see the presentation.
All right. Okay. Yeah, you cannot see, of course, because I didn't share. Now, of course, yeah. All right. Now you should see the presentation.
No, perfect.
Okay, sorry
Thank
for
you.
that.
No worries.
Yeah, just to see the people that are working with me. Yeah, no, that is, yeah. Okay. Setting the scene. What is phenomics in the title? Phenomics is emerging as a major new technical discipline in biology. And we define it in the cost action as an ensemble of methodologies and technologies for the acquisition, analysis, and the exploitation of high-dimensional phenotypic data on an organism-wide scale. So this is a definition that we developed through the cost action. And we will do our best to put also in Wikipedia. We'll see what will happen. But simplifying what is phenomics is going deep into the phenotyping. In animal science, phenotyping is the key. And that's the reason because we call the dogma in some way on animal science because it takes really the pivotal role in this field. Then another word of my title, precision. I went to the dictionaries because I'm not a native speaker, English person. And I take the definition of precision, the quality or state of being precise, exactness, and the degree of refinement with which an operation is performed, or the measurement state, accuracy, exactness and accuracy. So in this context, we need to improve exactness in our phenotyping approach and accuracy in the measurement at different levels, at different time points. So this is a key point. And for true precision stories, true precision sites of the same field, animal science, where livestock farming could become precision livestock farming, and animal breeding and selection could be defined by this new acronym that is derived from the first letters of these words: precision, animal breeding and selection. Why we need to go also in the precision side in animal selection? I borrowed this slide from EFAB, the European Forum of Farm Animal Breeders, because the breeding objectives have been changed over the time, over the period that go from the 70s, the last century, till today. So breeding objectives are changed, so we need additional phenotypes, more accuracy in measuring the animals for many different purposes. Nowadays, we have to improve animal welfare and health and welfare. We aim to better production and quality of the products, better use of resources, reduction of environmental impact. We need to ensure food security for many different concepts. And of course, we need to also maintain genetic diversity. So we need to have additional phenotyping approaches in this context, and more phenotypes are needed because so the final estimated breeding value, global estimated breeding value, derived from many different parts of the production aims of the animals. But of course, we have now a bottleneck. Go back to this concept. We have a major bottleneck in PLF and PABS, precision animal breeding and selection. Phenotyping is a limiting factor, actually, as we stay on what we already have. I mean, so we cannot go in the direction of PLF, precision animal breeding and selection, if we do not find or develop or understand new phenotypes. We need to collect new phenotypes. We need to identify new way to measure even all phenotypes in new way, in precise way. And we need to use in a novel way the phenotypes. So this is the limiting factor of what we have right now. And this is clear. And that's the reason because we developed the cost action that I'm chairing. European Network on Livestock Phenomics started in 2023. Will last for additional 2 years. We'll end on 2027 with the possibility to extend for 1 year. For who don't know what is the cost action, these are just a few concepts. Cost action is not a research project. It is a network. It is a research network. It is the interdisciplinary research network that brings researcher innovators Innovators means people that are not only scientists, but also employed in industries, are stakeholders, policymakers, decision makers, and so on. We need to bring them together to investigate a topic of our choice, actually, for 4 years. And this is the topic that we selected, Livestock Phenomics. The cost action are made up by many partners, actually, many individual partners that are representing the organizations. Cost action offer an inclusive pan-European environment for individuals of all levels of seniority to grow their professional research network and boost their careers. Pan-European environments means it is not only close to European countries, is open to all countries in the world. So all interested people, scientists, stakeholders can join the cost action. Even if it is European-centered, it doesn't mean that it's closed only to for Europeans. What is the content of the cost action that we develop, a European network on livestock phenomics? Okay, we are here, of course. We want to overcome the major bottleneck in PLF and precision animal breeding and selection. That is what we discussed before. And we see an added value in combining PLF and precision animal breeding and selection. So animal breeders would take advantage from what PLF scientists or stakeholders or farmers have developed in this context, and vice versa. So we need to use the data for sort of dual purpose use of the data that have been collected. So that this is clear, because collecting data at the animal level is expensive. So we need to extract as much information as possible and make them useful for many purposes. The action challenge of our cost action is has been written in this way. Each word has a meaning, and we took a couple of weeks to develop what is our challenge. What are the challenges that we want to face? Foster the development, integration, organization, and practical implementation of technologies, tools, methods, approach, models, expertise, and resources useful to scan and interpret the animal phenome, to paving the way for novel scientific knowledge and application in the livestock production sector. We took quite a lot of time to develop this sentence, and that's the reason because I wanted to read here with you. And the subject of our activities is the animal phenome that is defined as the physical and molecular traits, the physical and molecular traits of an animal. We can also consider animal population, but of course, for breeding purposes, the single animal is the key element. Of course, we can classify the phenotypes in many different ways. I just put a few the type of a classification in these and the following slides. Animal phenotype can be classified according to the level in which they are measured. Cell, tissue, organ, and whole organism, the type of information that is recorded, the temporal acquisition of the information, one event, single event, a combination of events, a continuous monitoring. The objective of the collected parameters are different. What is the objective of collecting information? And they can be targeted, dynamic, stable, and so on. They could be also classified depending on the level of analysis, external or final phenotype, performance and morphological disease, or internal molecular phenotypes or endophenotype. So the final phenotype are determined by the contribution and interplay of many molecular phenotypes with a multi-level relationship and the interaction with environmental factors. This is the key issue also to be considered in this context. So what is the summary of this type of a classification? So we have a very broad heterogeneity in phenotypic classes, types. And that's the reason, because we need to understand them and to have a network and someone who would in some way take advantage from this diversity, but also to understand how to manage the diversity. And there is also a wide scientific array of approaches and technologies that can be used to capture and manage phenotypic information. So diversity, yes, complexity in capturing the information. And this is really important to understand. Complexity means many people, many specialists in this context. I cannot understand all technologies by myself. I need an expert that maybe are expert in digitalization of the movement of the animals or the behaviors or capturing information at the omics level and so on. Okay, you don't have to read all the content of the slide, but this is important to understand that cost action has research coordination objectives. Actually, we have 8 research coordination objectives, but also capacity building objectives. Capacity building means we want to develop and to spread the opportunity to do research in this context, not only in Europe, but also in many other countries. In Europe, we have a target specific, that means inclusiveness target countries that are the less advanced in terms of research outputs countries in Europe. So we have also a target to involve as many countries as possible in this direction. That means East European countries, Portugal, Greece, and other countries are included in this list. Capacity building means we want to involve also the young generation. That is important and is one of our target. Another element that I wanted to mention is the cost action is a networking tool that rely on research activities running in different groups. But of course, we have as a coordination action the need to coordinate research activities within countries and across groups in some way. So we have to get in touch with many people. So the structure of the cost action. I'm the chair, and I have the honor to be with the vice chair, Professor Thomas Norton. I am from the animal breeding side, and Thomas is a specialist in PLF. So we cover the 2 main sides of the cost action. We have 4 working groups, 64 managing committee members from 36 cost countries that are included in the list of the countries that can be included in the managing committee. This is the decision body of the cost action, but also the running activities are planned in the working groups. And we have at present more than 450 working group members from 51 countries. Okay, so this is me, and this is Thomas Norton from KU Leuven, Belgium. Yeah, this is the overview of the 51 countries that are represented in the working groups with a number of people included. I will go back to later on how to join the cost action. The cost action is divided in 4 key specific scientific working groups, plus another one that is across all other working groups. So we have working group 1, that is phenotyping technologies. So we are discussing how phenotyping technology can be improved, and I will go back to the details later. Working group 2, genome to phenome integration, that is the genetic part of the cost action. Then there is another working group that embraces the activities on the previous two, computational resource and methodologies for data analysis. Actually, phenomics is a big data discipline in some way. We produce tons of data. Just recording the behavior of animals means a gigabyte of storage. So it is clear that it's a big data. Then you need to extract the relevant information, and all activities around the methodologies for data analysis are quite relevant. Working group 4 is focused on economic impact, regulation, policy, and society. And we have working group 5 focused on stakeholder engagement, communication, and dissemination. And this is the number of people that are involved in the different working groups. Actually, we have more 453 working group members that have been divided in these 5 working groups. That means one working group member can be interested in more than one working group. So working group 1 is the most, in some way, crowded in terms of number of people. Yeah, and we have also a young researcher innovator committee because we want to also to integrate in the discussion the young generation, and these are the name of the people. What are our tools? We organize meeting workshops, and we want to give to young scientists mainly, but not only to them, but to scientists interested in moving from one lab to another one, money. We want to give money. We distribute money in some way. And money for free. What does it mean? It means that you need to apply to get the money, and if you propose something that is interesting, you are granted. And you can develop something, of course, aligned to the aim of the cost action. We are organizing training schools, short-term scientific missions. That means we are paying people to move in one lab to another one for 1 week, 2 weeks, 1 month, 2 months, depending on what is important. Virtual mobility grants are the money that are given to produce some activities virtually. That means people can stay at home, but working together on a specific topic that is aligned to the objective of the cost action. And then we have dissemination activities. Also, we have job placement announcement service, mid-test and colder meeting that has been just organized, and then we are organizing demonstration activities in this context. The objective of the cost action, we can go a little bit deeper on this context. The objectives are our deliverables. You don't have to actually to read everything, but we have deliverables. We need to produce some materials. For example, deliverable D1, the first deliverable working group 1, a document report with a list of phenotypic technology approaches that can be used in Livestock Phenomics, and we have the amount of deliverable. So even if we don't have specific money to do research, we need to produce some deliverables that are attached to research activity some way. One deliverable is quite interesting. We are developing a book on Livestock Phenomics that will be that will include basic and advanced chapters to benefit both expert and non-specialists. If you are interested in contributing, of course, the cost action is open to all contribution, and also the deliverables are open to all contribution. We are, of course, coordinating the production of deliverables within the working groups. Just to give some details on working group 1, the main aim is to provide an overview of current phenotypic technology infrastructure that can be used for application in Livestock Phenomics, define a roadmap of research needs to capture high-dimensional phenotypic information on an animal-wide scale. And we have tasks: phenotypic technology mapping, standardization of phenotyping system and information, R&D infrastructure focused on phenomics in animals, identification of technology gaps and research needs, working group training school, working group 1 meetings, and so on, that are running all the 4 years of the cost action. And then the details of the deliverables. So of course, yeah, it's just an example. Our cost action is structured. We don't have to go on all working groups. Of course, the details. So I scroll down everything. Okay, so just stop just here for working group 4 that I think is a key working group. Because we don't have just to do science in this context and do not pay attention to what could be the potential economic impact of what we are doing, how we can transfer the information, how accessible is the information, and how is the perception of phenomics applied in livestock. So we need to pay attention also to this aspect, the economic impact, what is the regulation behind what we are doing, the policies that can be developed or have been already developed, and what is the impact at the society level. So I think this working group is really important, and we need to identify additional people that are interested to join this working group. Yeah, okay. I skip some of the slides. The events and the activities, what we are doing, what we did. The cost action was born in 2023, and actually we had a kickoff meeting in September, but the first annual meeting was held last year in June in Bologna at my university, where we paired this event with the annual meeting of the EFAB, the European Forum of Farm Animal Breeders. We are trying to have organized meetings in parallel or together with other events in order to give the possibility to people to also attend a conference or other events. This enlarge, enhance our networking activities. We had a training school last year at ILVO in Belgium, focused on phenotyping technologies in animals. There were 20 people that we funded to go for a week in Belgium to work together on phenotyping technology, novel phenotyping technologies, to understand how to manage the systems, the tools, and so on. And also we are organizing also national labs. What does it mean, national labs? National labs means that we are trying to spread our activities and create mirror groups of our cost action, because we want to enter into the different countries, because sometimes people do not speak English, or maybe they have some problems, and we want to also enter the local level, because we want to establish networks that will last for a long period. So our vision is not just to complete the activities at the end of our construction, but also to give a long-term perspective of what we are doing. So we want to develop something that will last for a while after the money from the cost action will finish. And we started the constitution of national labs. We went in Czech Republic, and we are developing national labs in Italy, Czech Republic, as I mentioned. We have developed in Slovakia, and we are developing national labs in Belgium, UK, and Ireland, the Balkan region, in Turkey, Switzerland, and so on, in order to include as many people as possible into this direction in a coordination manner, in order to just take advantage from what has been developed in one country, another country, and transfer the knowledge, and also take advantage from what has been developed through projects that could be advanced or less advanced. This year we just came from the annual meeting. It was organized in Leuven, Belgium, last week. And we had a very interesting stakeholder meeting focused on tackling the phenotyping bottleneck in livestock genetics with great speakers. And we talked about digital phenotyping. We talked about the involvement in industries. And we also talked about collaboration between different groups and different European projects. And also we discussed the possibility to produce a white paper in collaboration with US scientists. We have a roadmap in this direction, and we are finalizing the white paper. The white paper will be drafted and will be available by probably the end of October for refinement, and we hope to present it if possibly the next PAG meeting as a first draft, and the final version at the World Congress next year in Madison. So we'll see if this roadmap will be effective. We are doing our best to finalize the white paper focus on animal phenomics in collaboration between US and the European scientists. If you are interested, please contact me, and I can provide information. And if you are interested in collaborating in writing some parts, of course, the door is open. We are now running a training school in Leuven. And at present, we have 18 trainees on site, 10 trainees online, and 10 trainers. And the summer school is from data to discovery, measuring, analyzing livestock phenotypes. And we are paying people to be on site, and very interesting training school that we are running right now. And these are the map of the activities that we have done, we have completed. So I will be also in Korea, in Daejeong, in South Korea, in a few days to present the cost action. And if you are there, feel free to approach me and discuss how to contribute to the cost action. And we are organizing a meeting in together at the EAP Congress in Innsbruck. We are organizing another event, Young Research Innovator meeting at the Animal Science Days in Nitra this year in Slovakia. And we are plan to organize many different activities and collaboration and networking activities next year. So if you have some ideas how to eventually contribute to the development of our network, please drop me an email. We also organizing EU-LI-PHE talks to present some of the topics, and also if you are interested, you can be enrolled. If you have some interesting topics to be presented, we can include also in our webinar series. And our outlook vision, final outlook vision is yes, to overcome the major bottleneck in animal science. And the hashtag that we are launching, Phenomic, as the central dogma in animal science, is our key message. And we need to also consider PLF and precision animal breeding and selection as contaminating subfields in the animal field, because they both need data, and they both need phenotypes to develop their concept, their activities. We have open calls all over the period. Budget period goes from the 1st of November to the 31st of October each year. And open calls means if you are interested in applying, go to the website EU-LI-PHE. You can just Google EU-LI-PHE cost action and you can find it. Open call for short-term mission, inclusiveness target country conference grants, Young Research Innovator conference grants and dissemination grants. What does it mean, conference grants? We pay people to go, we give some funds for people to go to conference and attend and present their own work, focus on the cost action. And you can join the cost action, and it's easy. You can just scan this barcode, and you would be redirected to the eCost system. You need just to register to the eCost system, that is the cost action portal of the European Union. And after that, you can apply. You can go to the. You can apply to become a member of a working group. It is an easy task. You can take 10 minutes or no more, and you can be involved in the cost action and receive the message, and you are updated to the activities that we are running, and you can be informed of what would be the next activities and next possibility to contribute. And you can also follow us on our social media, LinkedIn, Facebook, X, and so on. And of course, if you are interested to have more information, you can contact Thomas and I, and these are the email address. Just leave here, and in this way you can scan the QR code and try to join also on the social media what we are doing. And thank you for giving me the opportunity, and thank you for listening. I will be available for any question you may have. Thank you again.
Thank you so much, Luca. I think your presentation of the cost action was quite on point, and you show us a pretty good overlook. So now let's see if people have some questions. Otherwise, I have one curiosity. Okay, let me just break the ice. So you mentioned the wide array of scientific approaches and technologies involved in the animal phenotyping. And my question is, how do you envision the role of machine learning and maybe AI in extracting meaningful phenotypic traits from these high throughput data sources that we have in the PLF, so precision livestock farming?
Yeah, this is an interesting question, actually. We are working also in the direction to integrate expertise into our cost action, because this will be probably the next challenge that we will have. Tons of data, and then we need to find the right information within this ton data, and maybe also to interpret the data, and maybe to develop something based on this, on the data that we have. So artificial intelligence will be one of the key tools that is available. It will not be the only one. We need also expert in many other fields, but of course, computational resources and managing data means are key, of course. And we have a working group dedicated to this specific activity. So artificial intelligence has been included in our cost action as a potential tool to extract also information and understand how to develop some of the information needed for application in precision livestock farming and animal breeding. Yeah.
Okay, so I think that on this channel, a lot of people can maybe help you if they are connected, or they will see your presentation. Jose, I think, has a question, and then we have another one in the chat.
Okay. Yes, so I have one question regarding more about the processing of the data. As you know, this group is from NFCore, and we develop standard pipelines for processing data, and mainly NFCore has been focused on genomics, but we are trying to open into other disciplines. And one question that I have, because actually I have been working in analyzing behavioral data during my PhD. And one of the issues is that, I mean, you have a device, you get the data in one format, each device, even if it's you were saying, no, the data is heterogeneous, but even if you are dealing with the same kind of data, you are getting maybe from one device the data in one format and with some information that with another device is not providing. So how do you envision that this could be standardized to produce like the standard pipelines for analyzing the data and so?
Yeah, well, this is another big issue, of course, the heterogeneity of the outcomes from different technologies, tools, or systems that produce data. And it is very difficult to integrate information coming from different sources. So this is a big, very big issue that prevent the possibility to share data and to use sometimes data. Across groups, across systems, across technologies, and we have a specific task focused on working group 3 related to the development of a standardized system to manage the data. Of course, we need to be realistic at the end because technologies are developing in a very fast pace, and it is quite difficult to pass all the time to think about standardization. That is clear. We need probably to define a set of rules, standard rules or elements that could then facilitate the integration of the data. That is something that would make sense. Otherwise, would be almost impossible to just go and try to standardize everything and then obtain standards for everything that could be useful. Otherwise, we will spend too much time and we lose opportunities because technologies are changing. There are new providers of, let's say, sensor data, and so on. So this is another key issue. Sometimes in industry, industrial providers of or manufacturer of a sensor or of other technologies that are interesting are not willing to share the raw data, and this could be quite critical, of course. There is a great discussion around these topics, and we'll see if we can address a minimal requirement to facilitate the transfer or maybe the integration of the information.
Okay, thanks.
Okay, so there is the question from Dominic Rocha. I think the pronunciation is this one. That is thanking you for your nice talk and is asking, do you include molecular phenotyping, for example, RNA-based, into your cost action or just physiologic phenotyping?
No, we also include all omics levels. There are molecular phenotypes, internal phenotypes. So we consider the different levels of phenotyping. Internal, that means RNA-seq data, metabolomics. Actually, we are working on metabolomics in pigs and other species. So we integrate this type of information. And we want to integrate this type of information with external phenotypes. So this is one of the key questions that we want to answer, how to integrate different levels of phenotyping. Thank you.
Much. So I think that if there are no more questions, we can close the talk here.
Okay. So yes, then I will close the talk here. Thank you, Luca, for the very nice and informative talk.
Thank you for giving me the opportunity.
Yes. And yes, as I mentioned at the beginning, the talk is recorded. So if you want to revisit or share it, it will be in the NF Core YouTube channel. And just a reminder that we run meetings each Thursday, Wednesday of each month. Next month, we won't have a meeting because it's August and for the holiday season, but we will come back on September, and you can either join the Slack channel or NF Core, Animal Genomics, or the mailing list, and you can get all the information about the next meetings. So thank you everyone for joining. And just to mention that we have a peak of 40 people during the meeting, so I think it was a very nice attendance on this meeting. Thank you everyone.
Thank you.
Thank
Have a good
you.
summer. Bye.
You too. Ciao.
Bye-bye.
Ciao.